Hello, my lovely anatomists and physiologists. Michele Glass here. We're back. We're still in our chapter 13. We're still thinking about those protections to the central nervous system, and we're going to dig a little deeper on the meninges. So I introduced the meninges as connective tissue. It's going to wrap around and support the entire central nervous system, which remember includes the brain and the spinal cord. And again, this is that pattern that we're seeing in the body where you have connective tissues on the outside of your organs. And remember, it's doing it's like a paradox, right? It's doing two opposite things at the same time, right? It's both separating that organ from the rest of the body, but then also connecting that organ back in to the rest of the body. When we look at our meninges, we'll see that there's three key layers. And so the Dura Mater or Mater is the outermost. And this actually translates to tough mother. Tough mother. What we see with the Dura Mater is that it is this really thick, fibrous, white, tough connective tissue layer. It's going to anchor the CNS to either the inner surface of the cranium. And here we're going to see what are called dural folds. Or it's anchoring the spinal cord to the vertebral cavity. So when we're talking about the brain is being anchored by the Dura Mater to the cranium. And we'll see the dural folds will be a key piece of this. And when we're talking about the spinal cord, we'll see that the Dura Mater is helping to anchor to the vertebral cavity. And then we have what's called the Arachnoid Mater. And this is very thin, but also fibrous tissue. So this is our middle layer. And it's going to help to really encase the central nervous system. And the extensions of the Arachnoid Mater look kind of like spider webbish, which is how we're getting this kind of arachnid reference here. And then our Pia Mater is our, what's it called here? Tender mother. So holding with care here. This is our innermost layer. It's going to hold the blood vessels to the nervous tissue. So this layer is like a single or couple cells thick. It's thin, it's transparent, it's connected into the neural tissue. So if you're doing a dissection and you have blood vessels, for instance, over top of the brain or over top of the spinal cord, you could take a forceps or tweezer and pull up at that blood vessel, and you would actually have a little bit of ripping or tearing that would happen in order to actually pull the Pia Mater away or off of that neural tissue. Now, in addition to looking at these connective tissue layers, we also have some key spaces. So we have above the Dura Mater, we have what's called the Epidural Space. Now this is only in the spinal cord. And it's going to be full of adipose tissue primarily, but we'll also see, of course, blood vessels, and we'll get some areolar tissue here as well. This is going to be an important cushion for that spinal cord in that vertebral column, right? So this is helping to pad in between the bone and the spinal cord. So this is going to be an important stabilizer. Again, the epidural space does not exist in the cranial meninges, only in the spinal meninges. When we look between the Dura Mater and the Arachnoid Mater in a living human, we don't really see a space there, but you will see a space in between the arachnoid and the pia mater, and we call this the subarachnoid space. And this space will be present in both the cranial and the spinal meninges, so meaning that both the brain and the spinal cord level. This space is going to be full of fluid called cerebrospinal fluid, which we mentioned previously, and we're going to dig deeper in its own video. So we're going to see cerebrospinal fluid is located here, and it can be abbreviated as CSF. There's no space in between the pia and the neural tissue. Remember, I said you can actually pull at that and feel and hear like a ripping. Sometimes you'll see people reference a subdural space. And again, my understanding is we don't really see that in a living, healthy person, but you might see that, you know, if a specimen has, I guess, been out for a while, maybe we'll say it in that kind of phrasing. Let's dig a little deeper into these dural folds. And what I'm going to try to do is add a picture. So what we're looking at in this picture is that same picture that we've been using for a reference. So we have here our left hemisphere. So we have a sagittal cut of the brain, and we're seeing the cerebrum, we're seeing the diencephalon, we're seeing the brainstem, we're seeing the cerebellum. So let me go ahead and label a few of those structures, and we're going with kind of like big labels here. So here's your cerebrum, here's your cerebellum. I'll label this as the brain stem, and then here you have your diencephalon. And what we're seeing with these dural folds, I like to describe this as sort of like bubble wrap. So when you're getting your packages from Amazon or wherever, you know, mostly items will arrive in a box. And if you just stick that item in that box, there's all that kind of space in the box for that item to bounce around and get damaged. And so they add in that plastic puff of air, right? So you have the plastic wrap, and the job of the plastic wrap is to kind of like hold around the object and sort of stabilize the object in that box so that it doesn't have as much room to like bump around in. And also even as it does bump, it's bumping into this, you know, plastic wrapped air, right? And so it's protected, it's cushioned. So the dural folds are acting in this kind of way. The dural folds are part of the Dura Mater. Remember our tough mother, and it's going to be anchored to the cranium. So the Dura Mater itself has an anchor to the cranium, and then it hangs down. And there's 3 to pick out. So we're going to name them, and then we're going to try to add them into our drawing. So we have what's called the Fox Cerebri. We have what's called the Tentorium Cerebelli. And then we have what's called the Fox Cerebelli. So let's start with the Falx Cerebri. The Fox term there means sickle shaped, so like a crescent. And then cerebri is talking about the cerebrum. And what we'll see is that this extends between left and right hemispheres of the cerebrum in the longitudinal fissure. So the longitudinal fissure is actually going from the top of the cerebrum all the way down to the corpus callosum, is this tissue that connects the left and right hemispheres together. So that's going to actually be tissue connected. So the fissure goes all the way to the top of the corpus callosum. So what I'm going to do is try to maybe grab a highlighter color. I'm going to see if this pink will show up. And what we can think of as our Dura Mater is going to be on the outside of the cerebrum, but then hanging down here to this point of the corpus callosum. So we're going to use this kind of pink, I'll highlight in pink, Fox Cerebri. So we're going to use this kind of pink to imagine this dura hanging down. So if you have this dura hanging down, this dural fold hanging down, this is helping to hold the brain in place. It's preventing it from having such a loose cavity to bounce around in, right? It's helping to stabilize it. But we're doing this in a way without really compromising the amount of neural tissue that we can have. So that's super cool. Okay, the next one for us to talk about is Tentorium Cerebelli. And you should think here literally of a tent. That's how we're getting this name Tentorium. Let me see if I can do a better tent when we have like a flap. That's maybe better. Cerebelli is of course talking about the cerebellum. So the Tentorium Cerebelli is going to extend over top of the cerebellum. So we're basically in between cerebrum and cerebellum. And here, let's see if we can grab, let's see about maybe this, maybe this sort of like a reddish color. And so the Fox Cerebelli, the Tentorium Cerebelli, again, we're going from the Dura Mater on the outside, and now we're hanging into this space here over top of the cerebellum, in between the cerebrum and the cerebellum. So again, we're helping to really kind of anchor or limit the amount of movement that the brain has in the cranial cavity without compromising the total amount of neural tissue. And then our last one is called the Falx Cerebelli. So remember, Falx here is talking about sickle-shaped. Cerebelli is talking about the cerebellum. Remember that the cerebellum is like the baby cerebrum. So it also has left and right hemispheres. So we're going to see that the Falx Cerebelli extends between left and right hemispheres, but now we're talking about the cerebellum. And we didn't really name that space. And so here, I think I'm gonna grab, let's see if yellow shows out. So it would be pretty much like extending into this area here. So pretty much where we have our cut of the cerebellum is basically where we have our left and right hemispheres, and so we can label that here. All right, this is it for our dural folds. You might want to research some additional photos on the Google to help you visualize a little bit better. Of course, bring your questions to me. Stay tuned for a deeper dive into cerebrospinal fluid, and as always, take care of yourselves and each other.